Welcome to the deep dive. Today, we're diving right into the fundamentals, really exploring the building blocks of the body, the cell, and how cells group together into tissues.
Yeah, and we're drawing this all directly from Porth's Pathophysiology.
Exactly. Think of this as your foundational map.
It really is. Our mission today is to give you, the learner, a clear picture of how cells work, their structure, function, communication, how they get energy. Because honestly, understanding disease, altered health states, it almost always starts here, at the cellular level.
If something goes wrong with the body, chances are the root cause is cellular malfunction.
Pretty much.
Okay.
So we need to understand the normal before we can tackle the abnormal.
Right. So we'll start small with the cell's internal parts, the organelles. Then we'll look at the cell membrane, how things get in and out, the electrical side of things.
And then zoom out to see how cells specialize and organize into the four main tissue types that make up everything else.
Okay, let's get started. Part 1, the cell itself. We're talking eukaryotic cells, right? Humans.
Correct. Eukaryotic means they have these membrane-bound compartments inside the organelles, unlike, say, bacteria, which are prokaryotes.
And inside it's filled with this fluid protoplasm.
Yep, protoplasm. Mostly water, like 70–85%,
Yeah.
but also packed with proteins, lipids, carbs, and electrolytes. Crucially, the distribution isn't random.
Ah, right. You mentioned the electrolytes earlier.
Yes. You absolutely have to remember this. Potassium, magnesium, phosphate, way higher inside the cell. Sodium, chloride, calcium, much higher outside.
And that difference is key.
It's fundamental. That gradient, that imbalance is potential energy. The cell uses it constantly.
Got it. Okay, so the biggest organelle, the one you can usually see, the nucleus, the control center.
Exactly. It holds the DNA, the genetic blueprint, all the instructions for making everything the cell needs, especially proteins. Think of the gene for insulin, for example. It's housed right there.
And it's also where RNA gets made.
That's right. DNA holds the master plan, but RNA molecules are the working copies. You've got mRNA, the messenger, carrying instructions out. RNA forms part of the ribosomes, the protein factories, and tRNA brings the building blocks, the amino acids, to those factories.
So the instructions leave the nucleus via mRNA. Where do they go first?
To the ribosomes. These are the actual sites where proteins are synthesized, assembled according to the mRNA sequence.
And these ribosomes are often attached to another big network,
Yeah.
the endoplasmic reticulum, the ER.
Yeah, the ER is this extensive membrane system. You've got two types. Rough ER has ribosomes stuck to it.
Hence the rough.
Exactly. It makes proteins destined for secretion out of the cell, like insulin, or proteins that will become part of the cell membranes.
And the smooth ER.
No ribosome, so it looks smooth. Its job is different, making lipids, steroid hormones, and it's also a major storage site for calcium ions, which is super important, especially in muscle cells where it's called the sarcoplasmic reticulum.
Now, you mentioned things going wrong. What about ER stress?
Ah, yes. Pathophysiology link here. If proteins start misfolding or piling up inside the ER faster than it can handle, that's ER stress.
And that's bad news.
Definitely. The cell sees it as a major problem. It can trigger inflammation, even program cell death, apoptosis. And this isn't just theoretical. ER stress is directly implicated in conditions like inflammatory bowel disease, type 2 diabetes, even some muscle diseases.
Wow. So problems with protein folding can have really widespread consequences.
Absolutely. Okay, so proteins made in the ER, especially those for export, they often move next to the Golgi complex.
The packaging center.
Pretty much. The Golgi modifies, sorts, and packages substances coming from the ER. It might trim a protein, like cutting proinsulin into active insulin, then bundle it up into a secretory vesicle ready to be shipped out of the cell.
Sort of like quality control and shipping combined.
You got it. Now, for cleanup and recycling, we have the lysosomes.
The digestive system of the cell.
Right. These are sacs filled with digestive enzymes, and they maintain a very acidic environment inside, around pH 5. They handle two main tasks. Heterophagy is digesting stuff brought in from outside, like a bacterium engulfed by a white blood cell.
And autophagy.
That's self-eating,
Well,
digesting old, worn-out organelles from within the cell itself. It's common when cells shrink, a process called atrophy.
what happens if the lysosomes can't digest something?
Good question. Sometimes indigestible material remains, forming what are called residual bodies. In cells that live a long time, like neurons or heart muscle cells, this can accumulate as leukofusin, the age pigment, kind of like cellular rust.
And if the problem isn't undigested waste, but the lysosome itself, like missing an enzyme?
Then you get lysosomal storage disorders. Tay-Sachs disease is a devastating example. Lack of a specific enzyme, hexosaminidase A, means a fatty substance called GM2 ganglioside builds up, particularly in nerve cells.
Leading to severe neurological damage.
Tragically, yes. It highlights how one specific enzyme defect can wreck an entire system.
Okay, all this building, packaging, cleaning, it must take energy.
A lot of energy. And that brings us to the mitochondria, the power plants.
Where ATP is made.
Correct. Through cellular respiration, they convert nutrients, primarily glucose, into ATP, the main energy currency. And crucially, this process, the efficient one, needs oxygen.
Anything else special about mitochondria?
Well, they have their own DNA, mtDNA, inherited from the mother, and they're key regulators of apoptosis programmed cell death. Too little apoptosis can contribute to cancer. Too much can lead to neurodegenerative diseases. They're really at a crucial control point.
Holding this whole complex factory together, giving it shape and allowing movement, is the cytoskeleton.
Right, the cell's internal skeleton. It's a network of protein filaments. You have microtubules, made of tubulin. They act like highways for transport inside the cell, form cilia and flagella, and are essential for cell division, pulling chromosomes apart during mitosis.
Which is why some cancer drugs target them.
Exactly. Drugs like vinblastine bind to tubulin and stop microtubules from forming, thus halting cell division in rapidly dividing cancer cells.
What other filaments are there?
Microfilaments. The key players here are actin and myosin, especially in muscle cells, where they slide past each other to cause contraction. But they're also involved in cell shape changes and movement, like during endocytosis and exocytosis.
And can the cytoskeleton itself be involved in disease?
Oh, definitely. Think about Alzheimer's disease. The neurofibrillary tangles that choke neurons are essentially collapsed, abnormal cytoskeletal structures.
Okay, let's move from the inside to the boundary. The cell membrane, the gatekeeper.
A crucial structure. It's this fluid lipid bilayer, primarily phospholipids, separating the inside from the outside. It's semi-permeable, selective about what gets across.
Mostly lipids, but the proteins do the real work.
For the most part, yes. Especially the transmembrane proteins are integral proteins that span the entire membrane. These act as channels or carriers for substances that can't just slip through the lipid part.
And if these channels or carriers are faulty,
you get channelopathies. Cystic fibrosis is a classic, tragic example. A mutation in the gene for a chloride channel protein, CFTR.
Right, affecting salt and water balance across membranes.
Exactly, leading to thick mucus in the lungs, pancreas, and other organs. It perfectly illustrates how a single faulty protein in the membrane can cause a complex multi-system disease. Another example could be certain forms of diabetes mellitus, involving changes in glucose transporters, the GOUT proteins.
So how do things actually cross this membrane? Let's talk transport.
Okay, broadly two ways, passive and active. Passive transport doesn't require the cell to expend energy. Things move down their concentration or electrochemical gradient.
Like rolling downhill.
Exactly. Simple diffusion is for small lipid-soluble things like oxygen, CO2, alcohol, steroid hormones. They just pass right through the lipid bilayer.
What about water-soluble stuff?
They need help. Facilitated diffusion uses transport proteins, either channels or carriers, but it's still passive, still moving from high concentration to low. Glucose getting into most cells via GLUT transporters is a key example.
And water itself.
Moves by osmosis, which is essentially the diffusion of water across a semi-permeable membrane, usually through specific water channels called aquaporins. Water moves towards areas with higher solute concentration.
Okay, that's passive. What about active transport?
This requires energy, usually ATP, because you're moving substances against their gradient uphill.
The main player here is that pump you mentioned earlier.
The Na plus K plus NH-ATPase pump, absolutely central. It constantly uses ATP to pump 3 sodium ions out of the cell and 2 potassium ions in.
Maintaining those concentration gradients we talked about?
Precisely. It keeps sodium high outside and potassium high inside. This is vital not just for maintaining cell volume, but also for the electrical potential across the membrane.
And the clinical link here is huge, right? What happens if this pump fails?
Catastrophe. If ATP production stops, maybe due to lack of oxygen or blood flow during a stroke or heart attack, the pump fails.
So sodium rushes in.
Sodium floods into the cell down its gradient. Potassium leaks out. Water follows the sodium osmotically. The cell swells up and, well, it often leads to cell lysis and death. That pump is critical for survival.
Wow. Okay, is there another type of active transport?
Yes, secondary active transport. It doesn't use ATP directly. Instead, it uses the energy stored in the gradient of one substance, often sodium, thanks to the Na plus K plus pump, to move another substance against its gradient.
Like using the downhill movement of sodium to drag glucose uphill with it.
Exactly. That's how glucose is absorbed in the intestine and kidneys, for instance. It's a co-transport mechanism.
What about moving really big stuff, like whole particles or large molecules?
For that, the cell uses vesicular transport. Endocytosis is bringing things in. Pinocytosis is cell drinking of fluids. Phagocytosis is cell eating of large particles like bacteria. And receptor-mediated endocytosis is highly specific, using receptors to bind cargo like LDL cholesterol before bringing it in.
And exocytosis is the reverse.
Yes, getting things out. Secretory vesicles fuse with the cell membrane and release their contents outside. Think neurotransmitter release at a synapse, or insulin secretion from pancreatic beta cells.
Speaking of electrical potential, let's touch on membrane potentials, that charge difference across the membrane.
Right. Because of the ion gradients maintained by the Na plus K plus pump and the selective permeability of the membrane, especially to potassium at rest, the inside of the cell is typically negative relative to the outside. This is the resting membrane potential, RMP.
Usually around the middle of 70 millivolts for a neuron.
Something like that, yeah. The cell is polarized. Now, changes in this potential are how cells like neurons and muscles signal.
Depolarization.
That's when the inside becomes less negative, closer to zero, or even positive, usually caused by positive ions like sodium or calcium flowing in.
And repolarization or hyperpolarization?
Repolarization is returning towards the resting potential after depolarization. Hyperpolarization is becoming even more negative than rest, maybe due to chloride flowing in or potassium flowing out.
How does this relate to signaling?
Small local changes are called graded potentials. But if a depolarization reaches a certain threshold, it triggers an action potential, a rapid, large, all-or-nothing reversal of the membrane potential that travels down the cell. This involves voltage-gated sodium and potassium channels opening and closing in sequence.
The basis of nerve impulses and muscle contraction triggers.
Exactly. And it has direct clinical relevance. For example, pancreatic beta cell sends high blood glucose. This leads to changes that close potassium channels, causing depolarization. That depolarization opens voltage-gated calcium channels, calcium flows in, and triggers the exocytosis, the release of insulin vesicles.
A beautiful link between metabolism, ion channels, membrane potential, and hormone release.
It really ties a lot together.
Okay, we've covered the cell in detail. Now, how do these cells organize? Part 4, body tissues.
Right. During development, embryonic cells differentiate into about 200 specialized cell types. These cells then associate to form the 4 basic tissue types. Remember the embryonic layers: ectoderm, mesoderm, endoderm. Different tissues arise from these.
What are the 4 main types?
Epithelial, connective, muscle, and nervous tissue.
Let's start with epithelial. Covers surfaces, lines, cavities.
Yep. Think skin epidermis, lining of the digestive tract, glands. Key feature. They're vascular, no direct blood supply. They rely on diffusion from underlying connective tissue.
And they're tightly packed.
Very. Held together by specialized junctions. Tight junctions seal the space between cells, preventing leaks. Adhering junctions provide strong mechanical attachments, and gap junctions are channels allowing direct communication and ion flow between adjacent cells.
Like in the heart muscle.
Exactly. Crucial for coordinated contraction. Epithelia are classified by shape: squamous, flat, cuboidal cube, columnar tall, and layers, simple one layer, stratified, multiple layers. Pseudostratified, looks layered but isn't.
You mentioned a pathology link here earlier, smoker's cough.
Right. The normal lining of the large airways is ciliated, pseudostratified columnar epithelium. The cilia beat to move mucus up and out. Chronic irritation from smoke causes this to be replaced by tougher, stratified squamous epithelium.
Which doesn't have cilia.
Correct. So you lose the mucus escalator. The only way to clear the airways is forceful coughing. It's called metaplasia, one cell type changing to another, usually less specialized but more protective one.
Okay, next type. Connective tissue, the most common.
By far. It binds, supports, and connects other tissues. It's characterized by having abundant extracellular matrix, ECM, the stuff between the cells, made of protein fibers like collagen and elastin, plus ground substance.
What falls under connective tissue? It seems broad.
It is. Includes loose connective tissue, dense connective tissue like tendons and ligaments, adipose tissue, fat, cartilage, bone, and even blood. They all share the basic structure of cells embedded in an ECM.
Got it. Third type, muscle tissue, specialized for contraction.
Yes, using those actin and myosin filaments we mentioned. 3 subtypes: skeletal, cardiac, and smooth.
Skeletal is voluntary, attached to bones, looks striped.
Right, striated. The functional unit is the sarcomere. Contraction is triggered by calcium released from the sarcoplasmic reticulum. Importantly, mature skeletal muscle cells generally can't divide to replace themselves.
Cardiac muscle. Also striated, but involuntary, found only in the heart.
Correct. And its cells are connected by those intercalated discs, which contain gap junctions for rapid electrical coupling, ensuring the heart beats as a unit. It also contains a specific protein called troponin.
Ah, the heart attack marker.
Precisely. When cardiac muscle cells, cardiomyocytes, are damaged and die during a heart attack, they release troponin and specific enzymes, like creatine kinase MB, into the blood. Detecting elevated levels of these is key for diagnosis.
And smooth muscle.
Non-striated involuntary, found in the walls of hollow organs like the gut, bladder, uterus, and blood vessels. Its contractions are slower and can be sustained longer than skeletal muscle. It uses calmodulin instead of troponin to regulate contraction.
Okay, final tissue type, nervous tissue.
The communication network, composed of two main cell types, neurons and glial cells. Neurons are the stars they generate and conduct electrical signals, the action potentials.
And glial cells are the support crew.
You could say that. They provide structural support, nourishment, insulation, myelin sheath formed by oligodendrocytes in the central nervous system, and Schwann cells in the peripheral nervous system, and maintain the environment around neurons. Communication between neurons happens at synapses, usually via chemical neurotransmitters.
Woo! That covers the 4 basic tissue types built from all those complex cells we discussed earlier.
It's an amazing hierarchy, from organelles to tissues, all working together.
It really is. We've gone from the nucleus managing the genetic code to mitochondria making ATP, the membrane controlling traffic with pumps and channels, and finally how these units build epithelial barriers, connective frameworks, contractile muscle, and communication networks.
A journey from the micro to the slightly less micro.
Okay, as we wrap up, time for that provocative thought. You mentioned lysosomes and residual bodies like lipofuscin. What about tattoos?
Yeah. Think about it. Tattoo pigment is injected deep into the dermis, the connective tissue layer below the epithelium. Those pigment particles are foreign, and they're too large for simple breakdown.
So cells try to clean them up.
Macrophages, a type of immune cell in the connective tissue and a type of phagocyte, engulf the pigment particles. But they can't fully digest them with their lysosomes.
So the pigment just stays inside the macrophage, like a permanent residual body.
Largely, yes. The pigment gets trapped within the lysosomes of these long-lived macrophages. Some pigment might sit freely in the extracellular matrix too, but much of it is locked inside these cells, potentially for decades. That's why tattoos are so persistent. It's cellular storage, essentially.
Wow. It really shows the limits and the persistence of cellular processes. That macrophage holds on to that indigestible ink for potentially your whole life.
It's a great illustration of those lysosomal functions and limitations in action.
A fantastic way to tie it all together. It underscores how understanding these fundamental cell and tissue functions is key to grasping both health and disease.
Couldn't agree more.
Well, thank you for taking this deep dive into the cellular world with us today. Keep exploring, keep questioning, and keep learning.